Good morning, everyone, and thank you for being here today. I'm excited to present to you the topic, Dr. S.K. Singh, Ph.D., Analysis of Flavors Used in Formulations. In this session, we'll explore how flavors are analyzed and integrated into various formulations. We'll cover the key techniques, challenges, and recent advancements in this field. Let's get started and delve into the fascinating world of flavors and their analysis. Let's talk about flavors in formulations. They are added primarily to improve palatability, making the product more enjoyable to consume. They also serve to mask any unpleasant tastes or odors that might be present, which is especially important in medicinal or health-related products. Additionally, adding flavors can significantly enhance patient and consumer compliance, encouraging consistent use of the product. However, it's important to recognize that flavors are complex mixtures. They can contain volatile organic compounds, which give them their aroma and taste. Sometimes, residual solvents are present from the manufacturing process. And we can't forget about allergens or potentially toxic impurities that might be in the mixture. Because of this complexity, regulatory control and analytical testing become absolutely essential. Ensuring the safety and quality of these flavors isn't just a formality. It's a critical step in the overall formulation process to protect consumers and meet legal standards. This ties directly into the overall theme of quality assurance throughout our product development. Let's take a closer look at the applicable regulations that govern flavoring substances. First, in India, we have the FSSAI, which sets standards for food products through the Food Safety and Standards Act. This ensures that any flavoring substances used in food meet specific safety criteria. Moving on, the USP, NF, or United States Pharmacopoeia, National Formulary includes general chapters on flavoring substances, providing a comprehensive guideline for their quality and safety. Then, we have the European Pharmacopoeia, which also sets standards for flavoring agents used within Europe, emphasizing purity and safety. Internationally, the WHO and FAO collaborate on the Codex Alimentarius, which provides global food standards, including guidelines on flavoring substances. In the United States, the FDA regulates flavoring substances under Title 21 CFR Parts 172 and 182, focusing on substances that are generally recognized as safe, or GRAS. The key takeaway here is that only approved flavoring substances, those that have been thoroughly evaluated and deemed safe by these regulatory bodies, are permitted for use. This ensures consumer safety and maintains high-quality standards across the food industry. Connecting this to our overall theme, understanding these regulations helps us ensure compliance and uphold product integrity in every step of flavor formulation. Let's take a closer look at the classification of flavors. First, we have natural flavors. These are derived directly from natural sources, like essential oils, such as peppermint or orange oil, which are extracted from the plants. Then there are extracts, like vanilla extract, which is obtained through a process of soaking and distillation. Oleoresins are also included here. They are concentrated extracts that contain both the flavor and some of the natural aroma compounds from the source. Next, we have nature-identical flavors. These are a bit unique because they are chemically synthesized, but are identical to the compounds found naturally. So, although they are created in a lab, they match the natural flavors exactly, making them a popular choice for consistency and cost-effectiveness. Finally, there are artificial flavors. These are entirely synthesized compounds that do not occur naturally in any source. They are designed to mimic natural flavors, but are produced through chemical processes. Understanding these classifications helps us see how flavors can be sourced or created, which is essential in the overall food industry, especially when considering quality, cost, and consumer preferences. Now, let's take a closer look at the quality attributes of flavors, specifically focusing on what we call critical quality parameters, or CQPs. These are essential factors that determine the overall quality and safety of a flavor. First up is identity. This ensures that the flavor is what it's supposed to be, matching the intended profile. Next, we have organoleptic characteristics, which include the taste, aroma, and appearance, basically, how the flavor is perceived through our senses. Then there's the composition profile, which looks at the specific chemical makeup of the flavor, ensuring consistency from batch to batch. Residual solvents are also critical. They're the leftover solvents from manufacturing processes, and we need to keep these at safe levels. Heavy metals are another concern. Their presence must be monitored because they can be toxic. We also focus on specific marker compounds. These are signature chemicals that confirm the authenticity or quality of the flavor. And, where applicable, microbial quality is assessed to prevent any microbial contamination that could compromise safety or stability. All these parameters come together to ensure that the flavors we produce are not only appealing, but also safe and consistent, which ties directly into our overall goal of delivering high-quality products that meet regulatory standards and consumer expectations. Now, let's talk about sampling and sample preparation. The first step is to collect a representative sample from the formulation or raw flavor. It's crucial to ensure that the sample accurately reflects the entire batch, so our analysis will be meaningful. Next, we need to protect the sample from light, heat, and air, as these factors can alter its composition and affect our results. Once we have a stable sample, the next step is to dilute it using a suitable solvent, commonly hexane, methanol, or ethanol, depending on what we're analyzing. Proper dilution is important to bring the sample into a workable range for analysis. After dilution, we filter the solution through a 0.45 micron membrane to remove any particulate matter, ensuring that our sample is clear and free of potential interferences. Finally, we prepare both standard and sample solutions. This step is essential for accurate quantification, as the standards help us calibrate our instruments and ensure reliable results. Overall, careful sampling and preparation are the foundation of good analytical work, helping us obtain trustworthy data that supports quality control and research objectives. Let's take a closer look at the identification tests we use in our analysis. One of the primary techniques is gas chromatography, often combined with mass spectrometry, known as GC or GC-MS. This method allows us to separate and identify the different compounds present in a sample with high precision. A key part of this process is comparing the fingerprint of the sample with that of a reference standard. This comparison helps us verify the identity of the compounds, especially those responsible for aroma. When it comes to key aroma compounds, identifying them accurately is crucial because they significantly influence the overall sensory profile. Now, for our acceptance criteria, the retention time and the mass spectrum obtained from the sample need to match those of the standard. If they align, it confirms that we've correctly identified the compound. Overall, these identification tests ensure that our analysis is both accurate and reliable, supporting consistent quality and authentic characterization of our samples. Now, let's take a look at Slide 8, which focuses on assay and composition analysis. Here, we utilize techniques like GC, FID, and GC-MS. These are important tools for our analysis because they allow us to identify and quantify specific compounds within a sample. When we talk about quantification of marker compounds, we're essentially measuring how much of these key substances are present, which helps us determine quality and consistency. For example, using GC, FID, we detect compounds based on their flame ionization response, making it very effective for hydrocarbons. On the other hand, GC-MS provides a detailed identification by mass spectrometry, giving us both qualitative and quantitative data. These methods are essential in ensuring that our products meet the desired specifications and standards. Overall, they play a crucial role in characterizing the composition of our samples accurately and reliably. Today, I'll be discussing the residual solvent analysis, focusing on the method we use. We typically employ gas chromatography, or GC, for this purpose. This approach aligns with the guidelines set by ICH Q3C, which provides a standardized framework for residual solvent testing. The key point here is that GC is a highly sensitive and reliable technique for detecting and quantifying residual solvents in pharmaceutical products. It allows us to ensure that solvent levels are within acceptable limits, safeguarding both product quality and patient safety. Connecting this to our overall theme, proper residual solvent analysis is crucial for compliance and maintaining high-quality standards throughout the manufacturing process. Now, let's turn our attention to heavy metal and elemental impurity testing. When it comes to methods, two of the most common techniques are ICP-MS, which stands for Inductively Coupled Plasma Mass Spectrometry, and AAS, or Atomic Absorption Spectroscopy. Both are highly sensitive and reliable for detecting trace levels of metals. These methods are essential because they help ensure product safety and quality by accurately identifying contaminant levels. Typically, the limits for these impurities are set very low, often in the parts per billion range, depending on the specific metal and regulatory standards. Understanding these testing methods and their limits is crucial because it allows us to maintain the purity of our materials and meet strict safety guidelines. It's all about making sure our products are safe for consumers and compliant with regulations. Now, let's talk about allergen and prohibited substance testing. When it comes to prohibited or restricted substances, there are specific guidelines we need to follow. For example, diacetyl and coumarin are restricted substances, meaning their levels must be carefully controlled and monitored. On the other hand, safrole is not permitted at all, so it should not be present in our products. It's important to comply with the limits set by both Codex and FSSAI standards, which are the international and national benchmarks for food safety. When testing, if a prohibited substance is not detected at all, that's exactly what we want to see, meaning our product is within safe and acceptable limits. This testing process is crucial because it helps ensure our products are safe for consumers and meet all regulatory requirements. Staying vigilant about these substances supports the overall goal of maintaining high food safety standards in everything we produce. Let's delve into microbiological quality, which is especially critical for liquid and natural flavors. When we talk about microbiological quality, we're focusing on ensuring that these flavors are free from harmful microorganisms. This is vital because contaminated flavors can pose health risks and affect product stability. Primarily, the concern here is about bacteria, yeasts, and molds that can grow in liquids or natural extracts. They can spoil the product, alter its taste, or even cause foodborne illnesses. To maintain high microbiological quality, we need rigorous testing and control measures throughout the production process. It's about setting strict hygiene standards, proper sanitation, and maybe even using preservatives when necessary. Remember, good microbiological quality supports the overall safety and integrity of our flavors, aligning with our goal of delivering safe, high-quality products to consumers. It's a key part of the quality assurance chain that underpins everything else we do in flavor production. All right, let's walk through this example of a GC assay calculation. Here, we have the area of the standard, which is 850,000, and the area of our sample, which is 820,000. To determine the assay percentage, we divide the sample area by the standard area and then multiply by 100. So 820,000 divided by 850,000 gives us approximately 0.9647, and multiplying by 100, that's roughly 96.47%. This means our sample's assay value is 96.47%. And importantly, this result complies with the specifications we're aiming for. This example highlights how we can use GC data to quickly assess whether a sample meets quality standards, connecting back to our overall theme of ensuring accuracy and consistency in our analytical methods. Let's now discuss the importance of stability and compatibility in our formulations. It's crucial to monitor how the flavor degrades under different conditions, such as heat, light, and pH extremes. These factors can significantly impact the quality and shelf life of our product. Additionally, we need to evaluate how our formulation interacts with excipients. as these components can influence stability and overall performance. It's also essential to retest the product at defined stability intervals to ensure it maintains its desired qualities over time. By thoroughly assessing these aspects, we can better predict how our product will behave in real-world conditions and ensure consistent quality for our consumers. In conclusion, it's essential that flavors are not only appealing, but also thoroughly approved, safe, and carefully controlled throughout the process. Using tools like GC or GC, MS remains our primary analytical methods. They help us ensure that every batch meets the highest standards. Controlling heavy metals, solvents, and allergens is absolutely critical to protect consumers and maintain product integrity. When we prioritize compliance, we're not just following regulations, we're safeguarding patient safety and gaining regulatory acceptance. Remember, good taste should never come at the expense of safety. Ultimately, our goal is to deliver products that are both enjoyable and safe, upholding the trust our consumers place in us. If you need any further support or have questions after today, please don't hesitate to reach out. I'm here to help you succeed and ensure you have all the resources you need. Thank you so much for your time and attention. I truly appreciate your engagement throughout this presentation. Remember, we're in this together, and your success is our top priority. Thanks again, and I look forward to connecting with you soon.